Well, aloha and welcome back to Anatomy 151 here at Chaminade University. Today we will be delving into Unit 12, or nervous tissue. So we'll be talking about the nervous system, and the overview of the chapter is that we'll be talking about the nervous system and how it maintains homeostasis. And also we'll talk about the different branches of the nervous system and the different types of cells that are found in the nervous tissue. All right, so there's two major types of the nervous systems. We have the central nervous system and the peripheral nervous system. And the central nervous system is comprised of the brain and the spinal cord. Basically, once information leaves the spinal cord and heads out through the spinal nerves, etc., that's all going to become part of the peripheral nervous system. So the peripheral nervous system involves the cranial nerves, the spinal nerves. And the difference between these cranial nerves actually only pass through one or two of the cranial vertebrae, the cervical vertebrae. They don't actually make it through the spinal column itself. The spinal nerves are going to branch off through the rest of the spinal cord. And they have little, if you picture that these are kind of like a highway, this would be an area where it's a merge, right? This is a ganglia. This is where multiple sets of nerves are going to come out at once through the little vertebrae, and then they're going to branch off. We also have enteric plexus. These are nerves that are going to be found in the small intestines. They're going to innervate this submucosal layer here. And we have tactile or sensory receptors in the skin that are also part of our nervous system pathway. That's all the peripheral nervous system. All right, so the central nervous system, again, that's this brain and the spinal cord. All of the information coming out the central nervous system is depicted here with the red arrow. The information going back in is going to go in through the blue arrow. Basically, information comes into the central nervous system by way of somatic receptors and sensory neurons throughout the entire body. So that information goes into the central nervous system. And we also have information coming out that heads to those somatic motor neurons, then the form of voluntary motion, and which is what's going to move our skeletal muscle and allow us to move around. That's all part of the somatic nervous system, which is part of the peripheral nervous system, right? We also have the autonomic nervous system. The autonomic nervous system, again, goes back and forth between the peripheral nervous system and the central nervous system. And autonomic sensory receptors and sensory neurons are going to send information, again, through the spinal column into the brain, and then information comes back out to the autonomic nervous system. And this is involuntary. This is split into both sympathetic and parasympathetic divisions. And these are motor responses that you are not able to control yourself the way you're able to control, say, your skeletal motion. We have the smooth muscle, cardiac muscle, and the glands that are part of the autonomic nervous system. And then we also have the enteric nervous system. The enteric nervous system is basically also involuntary, and it's in your enteric plexus. That includes things like your smooth muscle, glands, and the endocrine cells of the gastrointestinal tract. And again, just like all of the others, it's also constantly going to be in contact with the central nervous system. So all of these major systems, somatic, autonomic, and enteric, although they're primarily in the peripheral nervous system, they all interact with by sending information to and receiving information from the central nervous system. All right, so we have multiple functions of the nervous system. The first is sensory perception. We want to be able to sense the changes in our environment through sensory receptors, right? This is your sense of sight, smell, touch, taste, right? Sound. We also have motor nervous system. It's going to allow us to respond to stimuli. So, for example, you touch a hot stove and you immediately withdraw your hand so that you don't do further tissue damage. And also integrative nervous system. And this is a little bit more complex. This is when we're going to integrate information that's coming in and then make decisions about going forward regarding what appropriate behavior or actions to be taking. So this is going to be a kind of a combination of the central and peripheral nervous system, where it's going to be taking information in, basically learning and adapting, and then making decisions, and perhaps making a different decision in the future based on whatever result occurred in the past. Okay, so let's take a second to review the histology of the nervous system. We'll talk about neurons and neuroglia. I always like to liken neurons to princesses. Neurons don't do anything for themselves. Neuroglia are the handmaids of the neurons, and there's a ton of different types of handmaids that all perform different functions. But what is a neuron? So we may have talked about this previously when we talked a little bit about skeletal muscle and innervation, but just focusing on a neuron itself, we have multiple different parts. This here is the cell body. Right? The cell body is basically going to include the nucleus and all of the organelles, the cytoplasm. It's what you would consider to be a normal cell in comparison to what you think of when you think of a cell, right? You've got Golgi apparatus and mitochondria, etc. But we also have specialty structures. Some of those specialty structures are going to include these dendrites out here. which are going to be receiving information from the peripheral environment. And you can see that's denoted by these little, I want to call them stud-like regions. They're basically going to be receiving information sometimes from other axons, from other neurons, and sometimes from the surrounding environment. And so we have this dendrite region, which is going to be receiving information, and then this axon region, which is going to be sending information. And axons can be extraordinarily long. That's why there's this little splice here, just to show you that we haven't depicted it as long as it could possibly be. Or some of the axons in your body are going to run all the way to your toes from your brain, or from your early spinal column. So these can be extraordinarily long cells. This is why we have specialty regions. So around these, the axon, we have a specialty type of cell. It's called a Schwann cell. And the Schwann cell is going to have basically wrap multiple times around the axon. And so it's depicted as multiple layers, but if you picture it more along the lines of something that's a piece of tape wrapped around electrical cord many times, that's actually what that's more like, and that's a myelin sheath. And that myelin sheath prevents the electrical charge from getting out of the axon. If you picture this as like an electrical cord, and you want to make sure that you have that plastic wrapping around the outside of the electrical cord, or it might become leaky and you might lose electrical charge, right? So that's what these Schwann cells are going to do. They're going to protect that axon. And then there's actually going to be little areas in between that are going to be areas where information can kind of leap. And we'll talk about that when we talk about how they get excited, but those are called nodes of Ranvier. They're regions in between the Schwann cells where an electrical charge can actually jump from one to the next. Okay, and then these are the axon terminals that have the synaptic end bulb where information is going to be released in the form of neurotransmitters. And they might be released directly next to, or likely to be released directly next to, the dendrites of another neuron to pass information on, or they could be released right next to, say, muscle, where they are going to cause that muscle to fire. So these are electrically excitable cells that are able to take information from point A and pass them long distances to points B. Okay, we classify neurons based on their structure, and we have different ways that we can classify them based on where their cell body is relative to the rest of the cell. So if it's multipolar, that means that our cell body is on one side with the dendrites on one side with a very long axon and axon terminals on the other side. And every time you see this little bridge, that means that this could go on for a very long time frame, and we just don't have room to depict it here. If we have a bipolar neuron, that's because we have a very long, so the dendrites actually have a very long region itself for the information to travel, and the cell body is more in the middle. Right? This trigger zone, I'm going to step back just a second because I feel like I missed something. This axon hillock I did not talk about. The axon hillock is found at the very edge of the cell body as we're heading into the axon. And that axon hillock is going to be where we're actually going to have like a trigger zone. This is where we're going to be starting the electrical potential. So information comes into the dendrites, and then that's going to get passed on in the form of electrical information along this axon. And this is going to get triggered here at this trigger zone. Now, going back to the classification, the multipolar neuron, again, the cell body on one side, axons completely on the other. In the bipolar neuron, we have the dendrites very far away, or much further away than in this case, for example, than the cell body, and then the information is going to get passed out from the cell body to the axon, which is on the other side, in which case the cell body is going to be kind of in the middle. Now, if we have a unipolar neuron, that means that the information is going to be going from the cell body to a juncture that then is going to separate out into dendrites on one end and axon terminals on the other. And this region is going to be very similar to an axon, even though information is headed this way. So information comes in from the dendrites, and it comes along regions that are very similar to an axon, but isn't considered an axon until it gets to the other side. And the cell body is actually in the center of this long region. Okay, so that's the difference between multipolar, bipolar, and unipolar neurons. Now, a specialty type of a neuron is a Purkinje cell. Purkinje cells have a different set of what they call dendritic branching than do pyramidal cells. And you call them pyramidal cells because they look like pyramids. And this was discovered, I believe, by a scientist named Purkinje, but that is just a kind of a leap of my imagination there. My point here is that we have differences in the dendritic branching. So if you look at the dendrites here, they have a much different pattern than do these dendrites here. So this might be dendrites that are taking information from a much larger surface area, but taking a smaller sampling. But this, for example, would innervate maybe a smaller area, but take a lot more information from that area. All right, so multiple classifications of neurons. They can be either sensory neurons, which are known as afferent neurons, that information is flowing inward from the whatever tissue is sensing it, whether it's external, like your skin, or internal, like your intestines. Information comes inward. But we also have motor neurons, which are going to be efferent neurons, and information is headed outward, and they're going to generally fire. And then we have inter or association neurons. Now let's talk about different ways that we can have our sensory receptors laid out, or different types of sensory receptors and how they are laid out. So they have different layers of, again, that what we would call dendritic branching. Here we have a corpuscle of touch. And we may remember this from the integumentary system when we talked about the skin. The corpuscular touch is a little bit different from a tactile epithelial cell. So tactile epithelial cells have multiple mechanical receptors underneath the skin, which means that you can get information from a large surface area, and that information is all going to fire and head out to the cell body. The difference between that and something like a corpuscle of touch is that a corpuscle of touch is in a smaller area, and it's more like a pressure receptor, right? It's not going to be as far up on the skin either, and it's going to be more limited in the area in which it's going to be triggered. Once information comes into either one of these, it's going to head through the trigger zone to the axon, and then the axon is going to send information out to the axon terminal. And again, in this case, the cell body is going to be found right in the middle, which is considered to be this unipolar setup. Okay, because the cell body actually branches off. It's got its own little driveway from the rest of the axon. Here's a laminated corpuscle, very similar to a corpuscle of touch. That's why it's called a corpuscle. It looks like a little seed. And that information, again, is going to be passed down until we get to the axon terminal. And then we also have a nociceptor. And this is like a pain receptor, or I guess maybe a temperature sort of receptor. And the nociceptor is going to have a similar setup to the mechanoreceptor, except this is mechanical, and this will be set up to receive electrochemical signals or biochemical signals, but it'll do the same thing. It'll head down the axon and then head out to the axon terminal. So all of these sensory receptors are meant to take information from point A and transfer it to point B. All right, so let's talk about neuroglia. These are the nurse cells, or the maid cells. These are the cells that take care of the princesses, and there's multiple different types of them that do a bunch of different things or specialty functions. To be very clear, they are not electrically excitable. That means that they do not fire, they do not get triggered, they do not induce muscle to fire. Basically what they do is they tend to the neurons. They take up about half the volume of the nervous system, so there's quite a lot of them, and they can multiply and divide much more readily than neurons. We have a total of 6 different types of neuroglia, 4 that are found in our central nervous system, that is to say our brain and our spinal column, and 2 that are found in our peripheral nervous system, let's say the ganglion, etc. So the neuroglia of the central nervous system look kind of like this. So we have several different things. Let's identify the neurons. These are the princesses. Come on. So here's the neurons here. Neurons are going to be receiving information from dendrites and then taking that information and sending it out through their axons. Those axons are wrapped with Schwann cells. Schwann cells have a myelin sheath, right? Schwann cells are a particular type of oligodendrocytes. So oligodendrocytes are going to be cells that are going to form the myelin sheath, that are going to surround and electrically insulate the axon. All right, we also have microglial cells. That one's depicted here in green, and here in same one in green. We also have astrocytes. Those are depicted in blue. We have both fibrous, and here's a protoplasmic astrocytes. And then we also, so yeah, those are the 4 types of neuroglia in the central nervous system. And this is depicting here the pia mater, which is going to be part of the getting into the brain itself. Or the spinal column as well. So here is the neuroglia of the peripheral nervous system, and we're talking about here that it's a specific type of oligodendrocyte. It's a Schwann cell. It's going to wrap around that axon with that myelin sheath, which is going to electrically insulate the axon. This is depicting the nodes of Ranvier, whereby an electrical charge can jump from one set to the next. You can consider this like, instead of watching the TV program and fast forward until you find the spot that you want, jumping 2 minutes ahead or 2 minutes ahead. The nodes of Ranvier are going to basically just be a fast forward mechanism. We also, let's see, you have the unmyelinated axons that run through here. Let's see, so there's a different type of neuron here. It's going to have an unmyelinated axon. So we still have the Schwann cells that are going to wrap around it, but instead of having the myelin sheath, the Schwann cells are still going to afford some sort of electrical insulation, but it's not going to be wrapped as tightly or have the multiple layers that you see with the myelin sheath. All right, this one here is a satellite cell. And here's the neuron cell body in the ganglion, the satellite cell itself, Schwann cells, again, nodes of Ranvier, and the axon found in the middle. So the myelin sheath is very important. Again, it's produced by Schwann cells and oligodendrocytes, and it surrounds the axons of the neurons. Schwann cells here have their own nucleus, their own cytoplasm, and it just starts to wrap around and wrap around and wrap around until we have just a layer of myelin. So we start to lose the rest of the cell matter inside there and just end up leaving this residual myelin sheath, which wraps around that that axon. And again, that's going to provide electrical insulation for the axon. All right, so if you were to take a section of the brain, this is the frontal section, but if you were to take it through multiple different layers of the spinal cord, you're going to see the same thing. You're going to see gray matter versus white matter. And in the brain, you're going to see that gray matter is on the outside, whereas white matter is on the inside. And the inverse is true in the spinal column. So in the spinal column, this is what you're going to classically see as a spinal butterfly. Basically, what you're seeing is the gray matter in a butterfly shape inside the white matter. So it's the opposite because the white matter is on the outside as opposed to the inside the way it is in the brain. All right, so how do neurons communicate with one another? And we touched on this a little bit when we talked about muscles, but I'm going to get a little bit more into detail today. And basically, it all comes down to electrical excitability via something called an action potential or a graded potential. So action potentials basically allow communication over both short distances and long distances, whereas graded potentials are only going to be for short distances. But producing either one of them depends on changing the electrical charge of a membrane. So you have a resting membrane potential, and you have ion channels that are able to quickly flood positive ions in, and that's going to change the electrical charge of a membrane, or the electrical gradient, I guess, from one side of the membrane to the other, I guess is more accurate. Okay, another thing that's interesting about the brain and the spinal column is that they're going to switch. So as you are going to use the left side of your brain, it's going to be in control of the right side of your body, and the right side of your brain in control of the left side. We know that information comes from the brain through the central nervous system out to the peripheral nervous system. So when you see it shown here, it's going to be in the central nervous system from the brain going down through the spinal column until it hits a region of the spinal cord where it's going to exit out. Here's a ganglion, which is going to have basically a major intersection, or an exit off of a highway, if you will. That information is then going to be passed through from a sensory neuron from that point all the way down to the sensory receptor. Information then comes back up in the form of, information comes from the sensory receptor, up into the brain, and then comes back out where it's going to fire some of the action potentials into the skeletal muscle, allowing you to move. All right, so that's essentially how it works. Information comes in, depicted here is the corpuscle of touch going up as information that heads through the ganglia or that superhighway. Here's a connection between that sensory neuron and the information that's now headed into the central nervous system. It's going to go up into the brain, get passed back out from the brain in the form of electrical action potential that's going to get passed down into via the neuromuscular junction into the muscles themselves, firing the skeletal muscles and allowing you to move that pencil. Okay, but let's get a little bit more molecular. How does the action potential actually get started? An action potential happens because of ion channels. And ion channels are a specialty type of membrane structure that allow particular ions in. In this case, it's a potassium channel. When it's closed, potassium stays on one side. When it's open, potassium can leak out, right? We have a lot of potassium channels. They're a lot more numerous than, say, sodium channels. And these leakage channels basically alternate back and forth between being open and being closed. And that's going to allow for equilibrium to be restored. So we're going to end up getting approximately the same amount on one side and the other over time until we want to induce a change in the charge. We can also use ligands, and ligands are going to bind to receptors, if you remember your basic molecular biology. So ligands are going to be things that are going to be traveling through a liquid interface and then bind to receptors, which are bound to the membrane. In this case, the receptors are going to be found in the form of a channel. When the ligand binds, that's a chemical stimulus that induces the conformational change that opens the channel. Once the ligand-gated channel is open, then things can travel whichever direction they're intending to. We also have mechanical channels, so they're mechanically gated. So instead of having a ligand that's going to change it, we have a mechanical stimulus that's going to change it. And that depends on what mechanical stimulus instead of the chemical stimulus, I guess. And whatever it is that's going to induce that change, once it's open, then the molecules can flow, in or out, depending. We also have voltage-gated channels. So all of these are just different ways in which we can induce molecules to go back and forth across the membrane. Now, a voltage channel responds to a change in membrane potential. What is the membrane potential? Membrane potential is the difference between the charge on this side of the membrane and the charge on that side of the membrane. And that's called an electrochemical gradient or electric gradient. And if we have a change in that gradient, then we can induce this action potential. But I digress. So when we have a change in membrane potential, that's going to cause this channel to open. And when that channel opens up, that's going to, in this case, allow the potassium out. All right? So lots of different ways in which we can induce channels to open or close, which means that we can use different molecular mechanisms to target different channels at different points in these cycles. And that's going to become important as we move forward. All right, again, so leak channels are, again, channels that are just going to randomly open and close. They're found almost all over your cells, and that's going to allow for molecules to reach equilibrium in between different action potentials. We also have ligand-gated channels, or channels that respond to a chemical stimulus whereby a ligand from the outside environment binds to a receptor on the cell. We have mechanically gated channels, that they're going to respond to a mechanical stimulus, touch, pressure, vibration, stretching of the tissue, for example, or something that responds to an electrical charge, like a voltage-gated channel. All right, so let's talk about membrane potential. I just talked about that it all comes down to the difference in charge from the inside and the outside of the membrane. And that's true. So what it determines what we call the resting membrane potential, or the difference in charge that happens at a resting state. So in order for us to have a difference in charge, we have to have an unequal distribution of ions. And that along with the selective permeability to both of these are both positively charged molecules, right? So when we think about these molecules, you can't just think of it as a sodium ion or a potassium ion. You have to think of it as a positive charge, right? And so in terms of charge, we can switch them off interchangeably, right? So we can swap we can swap a potassium for a sodium and still have one positive charge. But only one can go through certain channels, and so we're able to regulate how much of what charge we have when. Anions, on the other hand, are not able to leave the cell. What do I mean by anions? Okay, cations are positively charged, anions are negatively charged. Right, but potassium and sodium, they're able to leave by multiple different ways, channels and pumps. Okay, so at a resting state, we are going to have a electrical potential or difference across the plasma membrane, and that resting state is approximately -70. What does that mean? That means that we have a little bit more of a negative charge in the cytosol, with say 70 millivolts worth of a negative charge in the cytosol inside the cell than we do in the extracellular fluid. So we have a little bit more of a positive charge in the extracellular fluid. And how do we measure that? We measure that with a specialty device called a microelectrode that's going to embed itself into the membrane and measure the electrical charge on this side versus that side. And this way we can record the membrane potential before, during, and after electrical impulses, i.e. action potentials. All right, so other things that contribute to the resting membrane potential, the percentages of different ions on either side. So chloride is a negative ion, sodium is a positive ion, phosphate, negative, potassium, positive, right? So we have different combinations of concentrations of all of these on either side of the membrane. We also have different types of channels. This is a leakage channel that only leaks out potassium, a leakage channel that only leaks in sodium, right? And then this is a specialty type of pump called a sodium-potassium pump. It requires ATP, but by using ATP, we're able to pump across 3 sodium and 2 potassium at the same time. It costs us cellular energy, but in this way, we're able to pump molecules from one side to the other. This lets us have a very tight regulation of our concentration of sodium and potassium on either side of the membrane. Okay, now, if we have a small deviation in that resting membrane potential, right, then we can end up changing the actual, basically we can end up changing the potential of the membrane. If we end up with a little bit longer of a membrane potential, we can end up with a depolarization event. What is a depolarization event? All right, so hyperpolarization and depolarization are equal and opposite sides of the same coin. Well, maybe not equal, I guess it depends on the charge. But a hyperpolarization event is when we end up more polarized. We end up with more of a negative charge than our normal resting potential. So here's our resting membrane potential at negative 70. Hyperpolarization means that we end up a little bit more negative. Like here's negative 73 or 75, it looks like. And if we end up a little bit more positive, that's a depolarization, right? So that's going to be an increase on this this millivolts, basically increase a little bit closer to positive. Now, anytime that we have the opening of either mechanically gated, ligand gated, etc. ion channel, all of a sudden a potential occurs. What does that mean? That means that we have a difference in charge between one side and the other, and we can change that charge based on the molecules that are on one side or the other. And we can look at the graded potential and how it reacts to the strength of the stimulus, or whatever the stimulus is, depending on the stimulus strength. So here we have three different stimuli that have three different changes in the membrane potential. And there's a direct correlation to the stimulus strength. So stimulus 1 is a weak stimulus, stimulus 2 a moderate stimulus, stimulus 3 a stronger stimulus. And a weak stimulus gives a weak change, right? We go from negative 70 to negative 65-ish. We go from negative 70 to negative 60 with the stronger stimulus. And with the strongest stimulus, we get the greatest change. We go from negative 70 to negative 55. So the amplitude of the potential depends on the stimulus strength, i.e. the change in the membrane electrical gradient depends on how strong of a stimulus you hit that membrane with. Additionally, if you hit the membrane with a stimulus that's very close to the first stimulus, so you hit them in rapid succession, you can actually double or increase the amplitude by adding them together. So if we have stimulus 1 that occurs prior, we get a change in the membrane potential, and then stimulus 2 occurs prior to allowing stimulus 1 to completely reach its regular resting membrane potential, we can have a summation or kind of an additive effect. So we can increase the change in membrane potential by having a second stimulus occur before the first stimulus's response is completed. All right, so that leads us to action potentials. So action potentials occur when we have many events occurring all at once that are going to lead to the decrease and then a reversal of the membrane potential. That's what we call depolarization. And depolarization is anytime we're depicted as an increase. So depolarization is shown as an increase because it's getting closer to 0. But if you look at the negative, it's going from negative 70 to negative 65. So it's going to be an increase in the membrane millivolts. But because we start negative, the number is going to get lower. So just kind of keep that in mind. Students sometimes get confused. So that's what a depolarization event is, if it goes up on that chart. Eventually it restores to the resting state, which is when we have a repolarization event. Then we get back to where we started. Okay, so we have several different phases of this action potential. So negative 70 is our baseline. Okay? And negative 70 to negative 55 is considered underneath the threshold. So any stimulus that's going to make this trigger occur, it has to get past the negative 55 mark. Once it hits the negative 55 mark, then we end up, that's called the absolute refractory period, then we end up opening up sodium channels and potassium channels, right? And so that's here. Basically, first part of that period is the depolarizing phase is when we're going to open up those voltage-gated sodium channel. And then that second part here, shown in this orange, well, I guess it's the red line, but it's shown in kind of a beige color, is going to be when we are opening up our potassium channels and our sodium channels are now inactivating. Right, so that's going to allow us to end up getting closer to where we were, right, when we want to be back at negative 70. For a moment, we actually dip underneath negative 70. That happens during our relative refractory period. And eventually, we end up returning back to our resting membrane potential. This is when we're able to fire again. During this phase, we're not able to get a secondary signal. But if we have multiple stimuli that occur right in this phase, we can head to this depolarizing phase faster. So those are the 5 major steps to the action potential. First, we start with our resting membrane potential. That's shown in purple here and also in purple here. Then shown in blue, we end up with a stimulus. Again, that stimulus has to reach a threshold, so we have to get above negative 55. Once we hit negative 54, boom, our sodium channels are going to open up. Once our sodium channels open up, that's going to drastically increase our millivolts until we actually end up in the positive range. We get to approximately positive 30, and then at that point, both our sodium channels close and our potassium channels open, and our millivolts are going to plummet. In this part here, this is our relative refractory period, during which time frame we actually going to dip down below negative 70, negative 75 or so, until we resume our resting membrane potential. Okay, so how do action potentials respond to stimulus strength? So I already told you that the stronger the stimulus, the increase in the action potential. But I also told you that we're able to induce multiple action potential, or one action potential from multiple stimuli if they come together in a very short time frame, in that form of summation that I spoke of earlier. Additionally, action potentials have to reach that certain threshold, right? This is not going to reach enough of a threshold to induce a stimulus. So a subthreshold stimulus, one that is too low, is going to cause a depolarization event, but not enough of a depolarization event to reach the threshold. Now this one is called a threshold stimulus. That means that we do have enough stimulus to reach that threshold to cause that action potential. Now if we have a stimulus that's stronger, that would be a suprathreshold stimulus. If it's stronger than this threshold, then we're able to induce this action potential several times, or sometimes it can be summative, all right? But in order for us to end up getting the action potential to be induced, we have to reach the threshold or exceed it. All right, so I talked about those channels and what they were doing at each stage. And if you'd like to pay attention here at the bottom of each figure, that's going to tell you what is happening at each stage. So that resting state is when we're at negative 70. Negative 70 is going to be the irregular electrochemical gradient from one side of the membrane to the other when we do not have any stimulus occurring. Okay? During this time frame, both your sodium and your calcium channels are, sodium and your potassium, I apologize, sodium and your potassium channels are closed. So both channels are closed. And we're at resting membrane potential. We have a small buildup of negative charges on the inside and a small buildup of positive charges on the outside. Again, that gives us a negative 70 until we get a stimulus. So once we end up with a stimulus that induces the threshold, so a subthreshold stimulus will not do this. We will depolarize and then return back to the resting state if it's subthreshold. But if we hit threshold, then we have an opening of the sodium channels. And when the sodium channels open, sodium ions are going to move through the channels, right? And that's going to cause a buildup of positive charges on the inside. So if sodium comes in, remember it was previously negative on the inside, sodium is going to flow in. Now we're going to end up with a bit of a positive charge. We're actually going to end up superseding 0, get a positive charge of approximately 30 until we're going to induce the repolarization phase. That's shown here in 3, and that's when we're showing that little red line that's going to start dropping again. Well, what happens biochemically at the membrane is that those sodium channels close and the potassium channels open. And potassium channels, remember, we're going the other direction, so now potassium is going to get pumped out. Once potassium pumps out, that's going to start making more of a positive charge on the outside, more of a negative charge on the inside, so restoring that negative charge till we eventually get to the point where we are getting all the way to the point where we've reached our original, we've actually dropped even further below that because we have gotten to this part here where we drop underneath our original resting membrane potential, and then we're going to eventually resume our resting membrane potential. And that happens as the repolarization phase continues. We're going to have continuous outflow of potassium, and as more potassium ions leave, the negative charges are going to build up inside the surface of the membrane again, and eventually we get to the point where we restore that negative 70. All right, so let's talk about the different types of action potentials. So we have graded potential, the different types of electrical induction that we can have. We have graded potentials and action potentials. All right, so the origin of the graded potential is that they're going to basically come from the dendrites and the cell body, whereas action potentials are going to arise from that trigger zone and then get fired all the way along the axon. So they're going to occur at different parts in the cell. Additionally, they're going to have different types of channels are going to be part of the graded potential or the action potential pathway. I apologize. Graded potentials are generally going to be ligand-gated or mechanically gated ion channels, whereas action potentials are generally going to be voltage-gated for sodium and potassium. Additionally, they differ in their conduction. So graded potentials are decremental. That means that they're not propagated. It means they're only going to be communicating from one cell to the next over short distances. However, action potentials are going to get propagated. That means they go from A to B to C to D to E to F. And that's going to permit communication over a much longer distance. And it also means that you can pass information on, say it went from A to both B and C, and then B and C both pass information on along two pathways. You can actually multiply information that's coming from one pathway and therefore cause multiple muscles to fire, for example. Also, the amplitude varies. So for graded potential, the amplitude is going to be directly dependent on the strength of the stimulus, whereas action potentials are pretty much all or none. Either it fires or it doesn't. So once you reach that threshold, you're going to induce firing. Now, the duration of graded potentials and action potentials differ as well, because graded potentials typically have a longer duration, oftentimes several milliseconds to several minutes, as opposed to just a millisecond or two for an action potential, half a millisecond to 2 milliseconds. So very quick firing. And polarity is also different. Graded potentials can sometimes be hyperpolarizing or depolarizing, whereas action potentials always have depolarizing, then repolarizing, and then a refractory period, and then that return to resting membrane potential. So they always have that characteristic graph that I just showed you, where we start at 70, we go up to -30, we drop down again a little bit below 70, and -70, and then we return back to -70. And in graded potentials, we have no refractory period at all. In fact, summation can occur, whereas summation does not occur with action potentials. So with graded potentials, a stronger stimulus or multiple stimuli that are going to be back to back to back can incur a stronger reaction. Whereas with an action potential, once you reach the threshold, you get the firing of the signal, and that is that. You cannot have a summation of signal. All right, so we also have two different types of conduction. So we can have continuous versus saltatory conduction. And this is what I was talking about when I was hinting at when I was talking about the nodes of Ranvier. So continuous conduction is basically when the flow along the axon is going to happen steadily in front of and behind, so that we're going to have this action potential flowing all the way down. And so what happens is we're going to have a constant cycling of this sodium. It's going to constantly going out and going in, going out and going in, going out and going in, which is going to then allow a change in the membrane potential, so that action potential is going to move from here to there. And this is a very A to B to C to D. This is watching the TV program in its entirety. However, we can also have saltatory conduction, whereby instead of traveling inside along the axon, we jump. And we do that by following one node of Ranvier to the next. You can picture this like a laser disc, right? Jumping to the next part of the TV program instead of having to watch all the way through. Oh, I've already seen this. And so you're able to just click, instead of fast forwarding through and waiting for each of them to be shown in one picture at a time, you're just jumping to the next sequence. And that's what's happening here. The sodium is going to, the sodium, which is inducing the change in the action potential, that's why it's called saltatory, it's going to be jumping forward through the nodes of Ranvier and basically skipping the intermediary parts of the axon. And it can be faster, as you can see here, 10 milliseconds in, and continuous conduction is only, what, a fifth of the way along. Here, 10 milliseconds in, and saltatory conduction has gotten much further, about 3 times as far. So it's a lot faster, and it relies on a specialty effect of these Schwann cells in that they're able to conduct that sodium around the outside. So instead of having, see here, we're cycling of the sodium in and out directly in front of, in and out, here we're going to have it actually flowing along the outside, and so we can have the electrical charge jump from one to the next. Okay, some of the factors that affect the propagation speed. What does that mean? How fast does that axon potential move from point A to point B, right? How fast does it propagate? One of the major things is the axon diameter. Secondarily, the amount of myelination. So how much myelin is there? The less myelin, we're going to have a little bit more leakage. And also temperature can play a role as well. Okay, let's talk about synapses. So synapses are regions between neurons. So where one neuron is passing on information to the next neuron, or where one neuron is passing information on to an effector cell, say a muscle cell, for example. So we can have electrical synapses, or we can have chemical synapses. Electrical synapses are going to synchronize the activity of multiple cells and allows information to transfer. It's going to allow information to transfer back and forth. A chemical synapse is only going to allow information to transfer one direction, from the presynaptic neuron to a postsynaptic neuron. Okay, this is kind of an overview of multiple neurons that are interacting and different ways that they can pass information along. So if we look at neuron, this yellow neuron here, as the neuron that is receiving the information, it can get that information from multiple ways. It can get it from the axon of another neuron interacting with the dendrites. That's called axodendritic. That's basically taking information that's passed on from axon A and receiving it from the dendrites of axon B, and then of course passing that information along to whatever axon is downstream. We can also have axosomatic. That means that the axon from this neuron is actually interacting with the cell body itself. So not with the dendrites, but with the somatic cell itself, the cell body. We can also have it interacting directly at the axon hillock. So that's called axoaxonic. So if you're getting information into the cell by the dendrites that comes from an axon of another one, it's called axodendritic. If you're getting information to the cell body from the axon of another neuron, that's called axosomatic. And if you're getting it from the axon of another axon directly here at the axon hillock, that's called axoaxonic. All right, this is the way that chemical information is passed at a synapse. So what is a synapse again? A synapse is a region where one neuron interacts with another neuron. And again, it can occur at multiple places, whether it occurs at the axon, the dendrite, or at the cell body. But what happens, we're taking this little region and we're going to blow it up. Here we have the presynaptic neuron. The nerve impulse comes down and it fires. When it fires, it's going to cause an influx of calcium. That influx of calcium then causes the release of these neurotransmitters. Usually that's acetylcholine. And when that release of the neurotransmitter occurs, it's going to get picked up by a receptor. The neurotransmitter receptor here is going to be closed until that ligand binds, and that ligand, again, that's acetylcholine. It's going to come out of this synaptic end bulb into the synaptic cleft, get picked up by the ligands here and cause the ligand-gated channel to open, and that's going to send information down and cause another nerve impulse. Okay, so we can have both excitatory and inhibitory postsynaptic potentials. And excitatory is basically when we're going to cause a depolarization potential, and inhibitory is when we cause a hyperpolarizing postsynaptic potential. Again, depolarizing is when we're going up and hyperpolarizing is when we are going down. And oftentimes a postsynaptic neuron is going to get information from many signals, and it's going to average it out and then decide whether or not to fire. So neurotransmitters have multiple structures, and we have ionotropic receptors and metabotropic receptors, and they can again cause either an excitatory or an inhibitory graded potential. All right, I'm going to skip over this because I'm not going to ask you specifically the differences between the different types of receptors. Now, the neurotransmitters that are arrived at that synaptic cleft can be removed multiple ways. They can be removed by diffusion or by degradation by enzymes, or they can be taken back up into the cell. So to summarize, if multiple presynaptic end bulbs release neurotransmitters, then it's going to be a combination of their effects. So if some are inhibitory and some are excitatory, then whichever one produces the bigger signal at the end is going to be the one that's going to have the nerve impulse. So it may or may not generate a nerve impulse due to the summation. Okay, so this is a type of spatial summation. So for example, it's the postsynaptic neuron. That means it's at the end of the synapse, and these guys are presynaptic. That means they're sending information. So their axons are interacting. In this case, it looks like they're interacting with the dendrites. And since both of them are sending the information in, we can either, if we have 1 and 2, and neither one of them by themselves are enough to cause the synapse to, or enough to cause the action potential to be passed on. But if we had both of them together, or many of them together, then we can perhaps reach that threshold level and induce that action potential. But again, it depends on how many of them are excitatory and how many of them are inhibitory. And once we get to the point where we can pass this threshold level, then we can induce that action potential. We can also have, so that's the difference between spatial summation and temporal summation. So spatial summation would be that we have multiple different neurons that are sending information to one postsynaptic neuron, and they're happening from different places, so spatially separated. Here we have temporal summation, and that means that we are releasing the signal at different time frames. So if this presynaptic neuron, just one, and postsynaptic neuron, again, just one, is releasing information every, say, 5 seconds, then it's not going to, or 5 milliseconds, it's not going to be enough. But if it starts releasing it closer and closer and closer together, then we can reach that threshold perhaps, and have temporal summation, where again we're going to induce that nerve action potential. So these are just different ways that we can have additive effects of information that's released at the synapses. So we can have information come from multiple different neurons that can add up, or we can have information coming from the same neuron multiple different times that adds up. And that's the difference between spatial and temporal summation. And again, this is just kind of an overview, but if you have multiple different presynaptic neurons that are sending information in to a postsynaptic neuron, then that information is all going to get added up, right? And whether that information causes a trigger has to do with the net summation of excitatory and inhibitory information coming in. Okay? Multiple different sets of neurotransmitters. Generally, for the purposes of our class, we'll be talking about acetylcholine, but there are other neurotransmitters that you may run into throughout the course of your college career. I'm not going to ask you about the chemical structure of any of them, but some of them include acetylcholine, nitric oxide, carbon monoxide, several different amino acids, including glutamate, aspartate, GABA, purines, and neuropeptides. Again, I'm not going to quiz you on any of the neuropeptides, but this is just a list of them if you'd like to further review them in the future. Some of the ones that you will be talking about later on, especially next semester, include these endorphins and CCK. And angiotensin 2, we'll also talk about this, is involved in the liquid balance, so the water regulation. Okay, so what's a neural circuit? So we just talked about one neuron passing information to another, or multiple neurons passing information on, but the entire pathway involves many different neurons, and they're all going to process information in a different way for a particular purpose. So we have several different types of circuits, including a simple series, a diverging series, a converging series, reverberating, and parallel after discharge. And basically what happens, if we're talking about diverging versus converging, you're talking about whether information is coming from one or two sources, then diverging out into many. So here we have one becoming two, becoming four. Or if it's converging, we have three here leading into one. And so the difference basically is, you have one that feeds into many, or do you have many that feeds into one? The difference between reverberating versus parallel after discharge is whether or not they're going to help regulate their own information. So basically, a reverberating circuit has a little bit of auto-regulation. So the information that comes from here gets passed here and then gets passed back up. Right? So that has a little bit of like a feedback loop. However, a parallel circuit is all going to have input one direction. So it's basically only feeding this way. It's never going to be regulating itself backwards. So that's a parallel after discharge versus reverberating circuits. All right. So let's talk about regeneration and repair. Nervous tissue is notoriously difficult to repair. Although it does have a little bit of plasticity, they have a very limited effect. Neurons have a very limited effect, the ability to regenerate themselves. So regeneration being defined as the ability to replicate or repair. So although they have what's called plasticity, which is the capability of change, they have very little regenerative capacity. In the central nervous system, there's very little to no repair that can occur, and that's going to be something like a spinal column break, etc. If we have influences from the neuroglia, including oligodendrocytes, for example, we have inhibitory influences from these regions, and we also have very few growth-stimulating cues that are present during fetal development that induce the differentiation of all the factors involved in the central nervous system. So it's one of those things that kind of has to be created while it's being created, and it really truly can't be fixed after it's already created. Another reason for that is because it induces rapid fibrosis, or formation of scar tissue after injury, and so that means that there's not a lot of space left for neuronal repair. The peripheral nervous system is a little bit more open to repair, so the peripheral nervous system repair is possible if the cell body is still intact, and additionally if the Schwann cells are still functional. So you have to picture that this is like an electrical cord, and the electrical tape is wrapped around the outside of it. Those are the Schwann cells, and if the Schwann cells are damaged, then the electrical cord is just going to be leaking electricity and is never going to be able to pass that information from point A to point B. However, if the cell body is still intact and the Schwann cells are still functional, and the scar tissue is not too extensive, then we can have a repair happening, which has three major steps. The first is something called chromatolysis, the second is Wallerian degeneration, and the last step is the formation of a regeneration tube. Now, I'm not going to quiz you on this, but I did leave this in because it is a very interesting mechanism of repair. It's a little bit unique. It is very unique specifically to this type of cells. So if we have damage, there's a normal neuron. So if we have damage that occurs, and we're going to have chromatolysis. That means it's going to be kind of broken down, and then Wallerian degeneration. So instead of just trying to repair the tissue, we basically start from scratch. We break it all down during this chromatolysis and Wallerian regeneration, and then we make a regeneration tube, and then that regeneration tube is going to eventually create the new nodes of Ranvier. So it's a very unique way of completely disassembling in order to remodel. All right, thank you very much for sticking with us today. I appreciate your time. Happy studying, and I will see you next time. Aloha.